the very kind introduction. So if we can get the first slide up, the title of my presentation is how we can incorporate proteomic and other omic data into clinical oncology. Many of you will be very familiar with the problems with treating cancer, and the first of these is that cancer is derived from our own cells, which leads to problems in finding treatments that distinguish between the cancer cells and normal cells. That's all I'll say about that particular problem in my presentation. And what I'll mostly focus on is the next problem, and that is that cancer is caused by multiple mutations in DNA. Typically, there is some form of genetic instability. There are many different causes of this, but the result is that there are multiple mutations in DNA. Many of them are random. That leads to heterogeneity within the cells within individual tumours, and it's a major source of treatment resistance. But a key outcome of this is every cancer is different from every other cancer, sometimes in subtle and sometimes in more major ways, which leads to the need to individualise or personalise treatment of cancer. Now, some of the recent breathless commentary about personalised treatment would lead you to think that this is a brand new idea in oncology. In fact, we've been individualising or personalising cancer treatment progressively for well over 100 years. Perhaps the first example of this was the use of oophorectomy by Beatson for treatment of breast cancer. And then, of course, histopathology is a very important determinant of personalised treatment. If you just take testicular cancer, for example, if the histology tells you it's a seminoma, you know that the treatment will be responsive to radiotherapy, often quite exquisitely. If it's a non-seminomatous germ cell tumour of the testis, then we've known for 40 years now that these tumours respond very well to platinum regimens. And then the third level of personalisation is with molecular data. And again, this is a concept which has been around and which is very familiar, I'm sure, to everybody in the audience. The use of molecular data such as estrogen receptor, so the presence of this single protein predicts that breast cancers will respond to anti-estrogens. And tamoxifen, the archetypal anti-estrogen, was approved for use in the UK back in 1972. So we have a very long history of personalised cancer treatment. What a lot of the recent interest in is the possibility of using molecular data for increasing personalisation of treatment. And of course, again, you'd be aware that there have been recent major advances in technology which allows us to look at massive amounts of molecular data, which goes by the generic term of omics. And of course, the archetype of that was genomics, and the genome, of course, is the sum total of all DNA in an organism. The epigenomics refers to the marks on DNA, which determine whether individual regions will be used. Transcriptomics refers to the sum total of RNA in a particular cell or tissue. Proteomics refers to the sum total of proteins in a cell or tissue. Glycomics, the sugar modifications, metabolomics, to small molecular metabolites, and so on. And without question, omics studies have completely transformed our understanding of the molecular basis of cancer. But what is lacking at the moment is a systematic approach to incorporating omics into clinical oncology practice. And that's what my talk is about. So understanding genomics of cancer is extremely important for understanding the causation of DNA, of cancer, because cancer is a disease of DNA, environmental causes, inherited mutations. This results through transcription and translation primarily in the output of proteins, which determine most of the functions of a living cell. And they also determine the responses, by and large, to treatment. So if we want to understand the causes of cancer, a lot of the emphasis has very rightly been on understanding DNA and less on proteins. But if we want to predict response to treatment, much less emphasis on DNA because proteins are closer to the action in terms of treatment. Because of the relative development of technologies and the explosion in DNA sequencing technology capabilities, which you'll hear a little bit, well, a lot more about from our third speaker this afternoon, John Christodoulou. DNA studies have been used as a surrogate for understanding responses to treatment by trying to predict what the proteins are going to be, essentially. But now it is possible, just in the last couple of years, to look at proteins on a high-throughput basis. This technology became available in the beginning of 2015, and with my co-conspirator, Professor Phil Robinson, who's a neuroscientist at Children's Medical Research Institute, but also has a very long history of expertise in proteomics, we've set about a project that we're calling the International Centre for the Proteome of Human Cancer. And the ACRF here stands for the Australian Cancer Research Foundation, which gave us a very major seed funding to get this going. And for short, we call it ProCan. So the concept of this project is extremely simple. And it's essentially that proteins and perhaps other omics can be used to predict what treatment a particular cancer is going to respond to. So the concept of the project is simply that for every newly diagnosed cancer, we intend to search a global database for the cancers with the most similar omics pattern. And again, keep in mind, we're predicting that the most useful information will come from the proteome, and then find out what treatments worked for those proteins, and just as importantly, what treatments didn't work. Now, that's a very simple concept, but of course, the global database has to be created, so the project is very complex. Now, why hasn't this been before? And I've hinted at this already, that it's technology-based or lack of technology till this point. Proteomics is not new. In fact, the word was coined here in Australia in 1994. But up until now, it hasn't been possible to study the proteome of cancer on a high-throughput basis. And the technologies that have been around for the last 15 years or so have required the use of quite large samples, and the results were not always reproducible from one lab to another. Using a new technology, our plan is to analyze the proteomes of 70,000 cancers of all types. So this is not just pediatric, but mostly adult, for which the outcome of treatment is already known, and to do this over the next 5 years so that we can build the global database that the outcome requires. And to do this, we're using a new and disruptive technology which was developed in Zurich in the lab of Professor Rudi Aebersold by a young postdoc, Tianwen Guo. And that was published with a very small series of renal cancers in Nature Medicine at the beginning of 2015. This is the technology that we've scaled up at CMRI in order to do this project. What it involves, just very briefly, is that very small samples can be taken. So we need a milligram or less, so that's a cubic millimeter or less of tumour. One of the key aspects of the technology is that the sample is then subjected to pressure cycling technology. So essentially, the sample is liquefied by cycling from 0 to 15 atmospheres in the presence of enzyme digestion. And then that goes through liquid chromatography and goes into a mass spectrometer using a particular type of mass spec analysis that we call swath MS. This produces a permanent digital record of the protein content of the tumour. So the features of this are that they're very small samples, it's highly reproducible, it's fast, we can do this within 24 hours, and it produces a complete record of proteins which can then be mined in the future. So if there's a particular protein that we become interested in 10 years' time, instead of having to do the analyses all over again, we can go back and mine the existing records. We've built what we call a data factory to produce this, so it's the 5th floor of our building here. The whole floor has been modelled so that it supports the workflow, and the key part of the factory is here, which is the mass spectrometry suite, and so far we have 6 very highly matched sets of equipment, all of which can produce the same quality data. So this is doing proteomics on an industrial scale, and it's one of only 2 industrialised proteomics centres globally, and it's the only one focused on cancers. So our timeline so far is that the PCT, so the pressure cycling technology, has been around for well over 10 years now. Swath MS has been around for more than 5 years. And what made this project possible has been bringing that combination together in the technology we call PCT Swath MS. So proof of principle at the beginning of 2015, we got a major award from the Australian Cancer Research Foundation to set this up in December 15. We built the facility and launched it in September last year, and we've started our first project. And we're on track, we believe, to doing 70,000 cancers over the next 5 to 7 years. So the outcomes are that within the next couple of years, we believe it will be possible to replace most of the existing protein-based cancer diagnostics, and most of the existing cancer diagnostics are in fact immunohistochemistries, which means that you're looking at one protein at a time, usually on a tumour section. We can measure many thousands of proteins simultaneously, and the immunohistochemistries which are in current use should be quite readily replaced by quantitative measurements from the mass spectrometers. And then when the database has been built to a sufficient size, we should be able to use computation-based both diagnostic and individualised treatment decision making for each individual patient. So this is the sort of thing we'd like to produce. I highlight here that we expect to be able to produce a report within a couple of days. In fact, at the moment, doing the analyses takes 24 hours. So this will fit very readily into a clinical workflow. We anticipate that a biopsy or an excised sample would go to the tissue pathology lab in the usual way, and at the same time that a sample is being prepared for histopathology, a milligram of tissue can be sent for proteomic analysis, and we should be able to have the complete report, the complete analysis done within 24 hours. So the sort of things that we anticipate having in the report would be a classification of the tumour, so the existing classifications and maybe additional classifications that are discovered through the course of the project. We should be able to report on all of the proteins which are currently of interest and important for making treatment decisions, and maybe there'll be additional individual proteins that we would report on. But most importantly, there will be an algorithm-driven prediction based on comparison of the protein pattern in this individual tumor with an increasingly large global database to make a prediction as to what treatment the tumor would respond to. So this will be faster than existing immunohistochemistry techniques. It should be more objective. Interpretation of immunohistochemistry requires a high level of skill. It may be cheaper than existing immunohistochemistry, and there should be an enhanced ability to predict the best treatment for individual patients. What we're talking about here is better deployment of existing treatments. But I think it's inconceivable that if we create a database of this size of new information about cancer, it's almost inconceivable that we won't discover new treatment targets, new molecules that can be used as the basis for developing new treatments. Now, in order to build a database of this size and to study enough tumors to make this really useful, we are needing to collaborate on both a national and an international scale. In order to get tumors of the type we need, we're anticipating that this will need about 100 at least different collaborating research groups, each of which has got a high level of expertise in a particular cancer type. The ideal collaborations are those where there's a very high level of clinical expertise. The absolute requirement for inclusion of tumors in this study is that the treatment outcomes are already known, or rarely prospective collections where the outcomes of treatment will be known very quickly. Ideally, the cohort would have been collected as part of a study for to address an important clinical question, for example, as part of a clinical trial. And importantly, so this is not an absolute requirement, but wherever possible, we're targeting tumor collections where other omic data are already available, especially whole genome sequencing. And I wish to emphasize, although what we're bringing to this project is a new and currently unique proteomics capability, in fact, this is a multi-omic study. And although we're guessing that most of the useful information will come from proteomics, we're not assuming that. That's a hypothesis to be tested. Okay, so what we have is the ProCan data factory, if you will, that supplies proteomics. We will add other omics capacity to it if the data justify that. And this will help build a global data and computational platform. So this will have omics data and the clinical data, especially treatment outcomes, and that will be the basis for algorithm-driven individualized treatment recommendations. And of course, we will welcome data from other sources. So if there are other high throughput proteomic facilities set up in the future, we will create this database in such a way that that data can be added as well, and of course other omics data. The way we are designing this, and what we would really like to see is that when this comes into routine clinical use, that all routine clinical data collected in the future would continue to feed this database. So with appropriate informed consent, any routine data of this type would be added to the database, and there would be routine outcome data collected, and we're working with the New South Wales health system to ensure that this occurs. So there'll be a two-way flow of information between the routine testing, which would use the global database for the treatment decision recommendation, but the outcome of that individual treatment would help to build the database for the future. And then this becomes a self-learning system because as clinical practice changes, as new drugs come into usage, it means that the database will be continually updated. So the skills of the core facility team that we have at the moment are particularly in proteomics, in anatomical pathology, in lab management, software engineering, bioinformatics, and health economics. These are the people who are or will be employed as core members of the team. And we have team leaders in place. So Tian Guo, who invented this technology in Zurich, has relocated to Sydney to be the scientific director of the project and to lead the proteomics factory. An anatomical pathologist, Rosemary Berlin, is leading the histopathology side of the project, and we've recruited from Oxford a lead software engineer, Brett Tully, and we're expecting to make our appointment of a lead bioinformatician in the near future. There are many opportunities for involvement of PhD students and clinical trainees, and in fact, medical oncologists, et cetera, in this. So, but particularly trainees in medical oncology, radiation oncology, surgical oncology, anatomical pathology, biochemical pathology, and clinical bioinformatics. So we're really actively looking for trainees who wish to be involved in this project. So that's a summary of what we're intending to do over the next 5 years, and I finish by acknowledging the sources of funding so far, which includes the Australian Cancer Research Foundation, which has really seeded this with a $10 million award for equipment. The New South Wales government, and in particular the Cancer Institute New South Wales, my own institute, Children's Medical Research Institute, and the Genes for Genes campaign, and the University of Sydney has also.